Hey, what's up, guys? It's me, Ali, one of your classmates in biology 111. All right. Okay, so I've studied through chapter 11 and chapter 11 through 17. Wasn't easy at all. So there was nights that I didn't sleep at all just because of the study. Not a big biology fan, to tell you the truth, but I try my best to learn it. Sometimes it gets interesting. One of the things that one of the topics that I didn't understand, well, it was a couple of topics that I didn't understand, is we're mainly on chapter 12. Let's see. One of them was Chargaff rules. That was really complicated. I guess I've learned it before, but I guess it slipped out of my head. And messenger RNA processing in eukaryotes. Eukaryote. I can't even pronounce it. Oh my gosh, I hate it. That's one of the reasons. One of the subjects that I quite understand much. Um, I actually went through a what's it called? What's it called? A tutor for biology on the at the academic skills center. So he actually broke it down to me for Chargaff rules and the messenger RNA processing eukaryotes. Almost said it. Okay, it's close. Um, One thing I learned about the Chargaff rules is that the the GNC and the ANT they connect. Well, they don't connect. G and A don't connect together in CNT. That's not what I mean. But ANT connects and the GNC connects. So the amount of A and T and G and C in the DNA varies from species to species. That's one thing I learned. In each species, the amount of A equals T, and the amount of G equals C. The DNA uses complementary bases pairing to store. That's what I meant. Genetic information. On each DNA, G and C is broken down to 20% G and 20% C. A and T is broken down to 30% A, 30% T. Sometimes you may see the U because sometimes it could be an RNA. It's just hard to tell from what I learned. Um, it can be GCCAU, GCCAT, GCCAUT. It's pretty much it can go so many ways. Just there's no words for it. Um, Another thing I learned is the messenger RNA processing in eukaryotes is that the DNA transcripts, which is translate the exon and introns towards the pre-RNA, and then it continues down to the 5 cap and 3 cap, the poly A tail. Um, there's another word that I can't really say. It's called spliciosum. Spliciosum said it. All right. It's called spliciosum. Um, that also transports, which is translate into the third row, fourth row of the diagram. Another thing is that once when it goes to the fourth row, it splits into the intro RNA, and then the pre-RNA slicing goes towards the nucleus and the cytoplasm. That's one thing. Got a little lost. I'm still trying to recuperate from that, but it's very interesting the way it breaks down because kind of gets my attention because I like to learn what it is. So another thing is too that I got a picture right here of the Eurychios and pro-Eurychios. Uh, there it goes right there. Pro-Eurychios, I think, is pretty much what's happened in the past. It's already done, and Eurychios is like present. Like there's DNA, then it transcripts to pre-RNA, the Cap4 to add, and then it removes the intron And then from the intron, the RNA, the mRNA leaves the nucleus, enters the cytoplasm, and then the mRNA translates the protein into the nucleus, which is inside, and it goes outside the cytoplasm. For the prokaryotes, the DNA transcripts the RNA, the mRNA, translating into proteins. That's the proeukaryotes. I'm sorry, guys, but I'm having trouble saying that word, that one word right there. The prokaryotes do not has a nucleus. The nucleus. I don't know if I'm saying it right or not. The nucleus. Just has a cytoplasm. That's one reason. One of the pictures right here I got, as I see. This is right here the eukaryotes, and this is the I can't even say it. Eukaryotes. Well, that's one of the things I learned, and pretty much that's what I'm going to talk about. Thank you guys, and I hope everybody passed their test. Thanks.